Good evening, everybody. Welcome to the BRLSI. This is a talk in the science series. I'm Stuart Reynolds, and I'm the convener of the science group of the BRLSI. And I should just begin with a housekeeping notice, which is that we don't do fire alarm practices on Thursday nights. So if the fire alarm goes, it's a real one, and we should leave the building immediately and assemble together somewhere in Queen Square. It will be obvious because there'll be lots of people milling around. There's a staircase which you came up to get here, and the simplest way to leave the building is down that staircase. Should it be difficult to go down that staircase, there's another staircase over there, and that will be available. So that's that out of the way. The other thing I'd like to say before we get started is that there are some other talks planned in the next couple of weeks that you might be interested in. So, for example, on November the 4th, we have a talk on, where are we? Here we go. It's actually a meeting of the Bath Naturalists Society, and it's about wasps. But if you're a biologist like me, then you quite like wasps. So good talk on wasps on November the 4th. And then on November the 7th, that's a Thursday, in the sustainability series of talks, there's going to be a talk about air pollution, particularly in relation to road traffic in cities, and that might interest some of you. But all that's in the future. And so now we can get on with this evening's event, which you've come to listen to. And we're very lucky to be able to welcome Dr. Ed Moody from Bristol University. Ed's a graduate from Portsmouth who migrated for more than one postgraduate degree from the University of Bristol in the Earth Sciences department, and he tells me that he's just migrated to the chemistry department in Bristol to do another postdoc. And I think this illustrates the point that so much of the really exciting advances in science turns out to be at the interface of more than one traditional scientific discipline. And perhaps we'll hear a bit about that in his talk tonight too. So as you can see from the title slide there, Ed is going to talk about LUCA, and I'm going to leave him to tell you what LUCA is.
Well, thank you very much, Stuart, and thanks for the great turnout, and thank you all for coming, and hopefully you'll learn a bit about the last universal common ancestor and life on Earth. So, let's say my name's Ed Moody, and I'm going to be talking about LUCA. There we go. Do I need to click something?
The arrow down.
There's a key on the laptop.
Oh, give it a second. I'll just do it like this. I'm going to do it like this. I'm going to do it, old-fashioned clicking the mouse. Yes, so, welcome to the talk. Life on Earth is incredibly diverse. So we have a whole host of beautiful different organisms living on Earth, from things like mammals to reptiles to amphibians to fish to plants to fungi, and a whole host of different unicellular organisms as well, like bacteria and archaea and a whole bunch of different eukaryotes as well. Life is also incredibly widespread. It's everywhere. It can inhabit a whole host of completely different environments, from sub-zero temperatures to above boiling point. But how did it begin? What was early life like? What was the early Earth like? These are hopefully some of the questions that I can try and answer over the course of my presentation. Not only is life diverse, widespread, it's also ancient. So the oldest kind of uncontroversial fossils, which people don't dispute are evidence for life, are about 3.4 billion years old, okay? So the Earth is around 4.5 billion years old. Oldest fossils, like this stromatolite here at the top, which is a kind of fossilized microbial mat, so layer upon layer upon layer of fossilized bacteria. About 3.4 billion years old. We've also got these kind of squills in the bottom right, which also represent really early life. There is evidence for life being slightly older than 3.4 billion years ago, but it's controversial. Not everyone agrees about it, and in order to really make claims about life much older than that, you really do have to have extraordinary proof. So we know that life is at least 3.4 billion years old, and probably younger than the Earth, which is about 4.5 billion years old. So not only is life old, diverse, related, widespread, it is also all related. All life on Earth shares a common ancestor at one point. And we know this through the conservation of fundamental biological characteristics, okay? You know this from the universal genetic code, the conservation of DNA to RNA to protein, the same set and chirality of 20 amino acids found across the tree of life, the same protein translation machinery, the ribosome, for example, and also the shared energy currency, so ATP, and the machinery associated with making ATP synthase. This is something shared across all domains of life. And one of the practical applications of this is that we can modify things like bacteria to make hormones like insulin for our own kind of benefit. So there are practical advantages to life, all life being related. So now I'm going to take you on a brief journey through our own evolutionary history. We're going to go down looking at various kind of common ancestors, right back to the origin of eukaryotes. So hopefully, we're all vaguely aware of our own species, Homo sapiens. It's thought that the common ancestor of Homo sapiens is about 300,000 years old. This is an estimate, so obviously some of these ages are very much subject to change, and there are going to be disagreements if you read various papers looking at the evolutionary history of humans. I think the oldest fossils are from about 230,000 years ago for humans, and using a combination of molecular clocks with these fossil evidence, we get an age of the common ancestor of sapiens about 300,000 years ago. So everyone in this room and everyone watching online is a Homo sapiens. We're going to go back further and look at the common ancestor of our genus. So the genus is Homo. We think the common ancestor of Homo is about 2.8 million years ago. Okay, so for an example of another member of the genus is the Neanderthal in the middle top here, the skeleton, and there's also various other ancient humans that you're probably aware of, like Homo habilis, Homo erectus, for example. On the bottom left here, I asked an AI to generate a reconstruction of the common ancestor of the genus Homo, and it came out with this thing, which looks fairly reasonable to me. It's not too bad. That looks like an ancient human, I think. I mean, I'm not an expert in ancient humans, but to me that looks vaguely correct. So we're going to push on backwards in time and see how other things look. Our family, Hominidae. So we think the common ancestor of hominids is about 17 million years old. These are also known as the great apes, and obviously it includes us, Neanderthals, but it also includes things like chimpanzees, bonobos, gorillas, orangutans. And here in the bottom left, we have another AI reconstruction, and this, at first glance, looks really convincing. This clearly looks like a great ape. But I was thinking about this, and actually, this is clearly like basically it looks like something from Planet of the Apes. It looks like a bipedal chimp, which is going to be massively incorrect because we don't think that the common ancestor of great apes is going to be a biped, right? This is probably quite wrong. I mean, you're going to have the hair. It looks quite ape-like, but again, probably a bit wrong, but not horrendously wrong, not too bad. Go back even further in time to about 64 million years ago is where the age of the common ancestor of primates is thought to have emerged. And this includes things like the old and New World monkeys, including all the great apes that I've mentioned, as well as things like lemurs. And so here, the common ancestor, again at first glance, looks kind of reasonable. It looks like a lemur-y, slow lorisy type thing. But actually, if you look closely, you can see that it's got two tails, which is obviously massively wrong. Probably. One of its arms is much bigger than the other. So overall, again, not really a great attempt. But I should say, I didn't alter this. I didn't update the reconstruction or anything. I just asked, give me the common ancestor of primates, and this is what it spat out. Go back even further in time to the common ancestor of all mammals. So this is estimated to be about 200 million years ago. And now this includes things like Afrotherians, things like elephants, chiropterans, bats, cetaceans, things like dolphins, armadillos, carnivorans like lions, tigers, cats, dogs, bears, etc. But also includes metatherians, the marsupials, things like kangaroo, Tasmanian devil, etc. And it also includes things like the platypus as well, and the echidna. And this reconstruction, I think it's pretty good. This looks pretty reasonable to me. This is probably what the common ancestor of mammals vaguely look like. I mean, it would have laid eggs. Obviously, you can't see that in the picture, but we do think it's probably some kind of rodent-looking type organism living around this time. We go back even further in time. So now, some of you may have noticed that I've kind of skipped a few steps. I've skipped things like the vertebrates, for example. So chordates includes obviously all vertebrates, including mammals, reptiles, amphibians, and all fish. But chordates also includes another couple of weird little groups. For example, the tunicates, so this kind of blobby thing in the middle here. These are tunicates, so these are actually quite closely related to, they're a sister group to vertebrates, as well as this lancelet here in the bottom right, which looks a bit like a wormy fish thing. So the reconstruction here is really, really quite wrong. So this, to me, looks a bit like a coelacanth or a sarcopterygian, a lobe-finned fish. We know that this would have come much later in time, common ancestor of sarcopterygians, which includes coelacanths, lungfishes, and tetrapods, things like mammals, reptiles, etc. And this reconstruction it clearly just looks like a coelacanth. We would certainly expect the common ancestor of all chordates, which their defining feature is a notochord. So it's not evolved a spine yet. It's more like a kind of rigid rod in the back of the organism. So it probably looked a lot more like this lancelet on the bottom right than anything like this coelacanth monster on the left. We go back even further in time to the common ancestor of all animals. So this now includes things like arthropods, including insects and crustaceans. You're going to include mollusks like squids, octopuses, snails, etc. Also non-bilaterian animals like cnidarians, such as jellyfish or coral, as well as things like sponges. So this is dated to about 665 million years ago. And this reconstruction, I like this. I think this looks good. I think this is probably roughly what the last common ancestor of animals looked like, essentially a sponge, or possibly some people would argue that the common ancestor is going to look more like a kind of a ctenophore, which is like a cube jellyfish. But that to me it's about right. Then we have a big time skip to the common ancestor of all eukaryotes. So this is 1.6 billion years old now. So this now doesn't only include just the animals, this also includes things like fungi, it includes things like the amoebas, but it also includes, on the other side of the eukaryotic tree, plants, all plant life. So the common ancestor here would have been a unicellular organism with a membrane bound, with a cell membrane. It would have had a nucleus, and it would have had a mitochondria. And so I'm not entirely sure what all this other stuff floating around in this ancient eukaryote is, but it's certainly interesting that this is what the AI has come up with. Its guess may be as good as anyone's. Okay, so I've introduced our domain of life, the eukaryotes, but now I'm going to introduce a couple of other domains of life to really set the context of this talk. So this takes us back in time to around I mean, I just said 1.6 billion years. This slightly outdated chart says around 2.2 billion years for the common ancestor of eukaryotes. So this kind of shows you how the time span where these estimates can kind of float around. So, still, if we go back further in time, we're going to go past the origin of photosynthesis. We're going to go past the late heavy bombardment. We're going to go past, or we're going to just leave it just after the formation of the moon, and we're going to talk about bacteria, okay? So probably all have heard of bacteria before, discovered in the 1600s, or at least seen for the first time in the 1600s by a Dutch scientist called Anthony van Leeuwenhoek. And he designed, made, and then used his own microscopes. So at the time, microscopes were limited to, I think, about 10 to 20 times magnification. He had this incredible technique where I think he polished and designed his own and made his own lenses, which allowed people for the first time to see almost 200 times. And so he looked at various things, including bacteria. He saw bacteria for the first time. Bacteria are a really fundamental component of everyday life. They're used in agriculture, but obviously in our own bodies as well, the microbiome, which scientists are slowly realizing how important that is. They play a massive part in the Earth's geochemical cycles, the carbon cycle, the nitrogen cycle, the sulfur cycle. These things are incredibly important. But as I'm sure you're all aware, they're also pathogenic. Now, we do share a common ancestor with bacteria, but it's slightly more complicated than just a direct kind of traditional Darwinian sense. We think bacteria actually gave rise to our mitochondria. So it's thought that a kind of floating cell engulfed a free-living alpha proteobacterium, and then this gave rise eventually to our mitochondria, where it lost a lot of genes, and now it just serves to help us generate energy. The host cell for that free-living bacterium was an archaea. So archaea are another domain of life, and these were discovered in the 70s by a pair, Carl Woese and George Fox. And these were initially thought to only live at incredibly extreme temperatures. So these were thought to live in kind of hydrothermal vents. All of them are thought to be methanogens. But actually, over the last couple of decades, we've found out that these things live everywhere, including in us. And although they look very similar to bacteria, I've possibly, to confuse you, I've included two very charismatic archaea in the bottom here, Pyrococcus furiosus on the left, as well as the newly cultured Asgard archaea, the Promethearchaeum on the bottom right. So these both, they're not your typical archaea. They have lots of dangly bits. Most archaea just look like your regular bacterium. But they're actually very different to bacteria. So their cell walls don't contain any peptidoglycan. Their cell membranes have a different way of the lipids being bonded to one another. And there's many other differences as well between archaea and bacteria. Interestingly, there aren't any known pathogenic archaea. So no one's ever found a pathogenic archaea, and we don't know of any archaea that actually hurt humans. So that's kind of interesting. So I'm going to take a brief aside now that I've introduced the kind of key players in the tree of life and talk about trees in general. Not those kind of trees, I'm going to talk about phylogenetic trees. So this is the field of study that really allows me to work back in time and look at the evolutionary history of different organisms. So in phylogenetics, we use the word tree to describe a hypothesis of evolutionary relationships, okay? Generally speaking, this is going to be different species. So on the bottom, in the middle here, we have a human assisted to chimpanzee. We've got lizard assisted to bird, and then the lizard bird assisted to human chimpanzee. These can also describe genes as well. So individual genes might have different evolutionary histories to those of species. So that's something to bear in mind. The way we write this on a computer, for example, in text is in the Newick format. So this is a series of brackets, the names of the taxa, and then commas and a colon. So this is where it gets slightly complicated, tree terminology, okay? So within a tree, you have a leaf, which seems fairly reasonable, or tip, and these are generally describing your sequences or your species, your taxa. These are separated by branches. Branches generally represent the amount of either real time or genetic distance between different species. And then you have nodes. So these are the internal nodes, and these are hypothetical ancestral lineages or organisms. Another thing that you have to bear in mind is that this tree on the left here is unrooted, okay? Which means there are kind of multiple different possible combinations that you can, that evolution may be taking. On the right here, we have a rooted tree. So the way we root a tree, generally speaking, is using an outgroup. All an outgroup is, it's an organism that's more distantly related to all the organisms that you're interested in, okay? So you take that as your outgroup and allows you to put a direction on the tree. The way we can talk about different groups is in terms of their kind of common ancestry, and we use the term monophyly to describe a group where you're including all descendants as well as their common ancestors. So this would be a monophyletic group. An example of that in real life, for example, would be primates. So primates includes things like lemurs, humans, and their common ancestors. This is a monophyletic group. But there are also groups which aren't monophyletic, which are used in everyday kind of language. For example, so paraphyletic groups, such as reptiles, okay? So when people use the term reptiles, generally speaking, they're going to be talking about scaly things which lay eggs, which seems fairly reasonable. But actually, genetically speaking, birds are much more closely related to crocodiles and dinosaurs than crocodiles and birds are to turtles or lizards, for example. So the everyday language use of the word reptiles would be a paraphyletic group. You know, we're not including all those the descendants there. And the actual kind of scientific, the taxonomic word for reptiles, including birds, would be saurians. So this would be the saurian clade. And then perhaps even more in a more complicated fashion, we have polyphyletic groups, okay? So these, I'm not sure off the top of my head of any common polyphyletic groups, but an example could be flying animals. So we could take something like butterflies and bats, and these are both flying animals, but there are many different kind of common ancestors and descendants which are more closely related to both butterflies and bats, which you wouldn't include in the flying animals group. So these are some of the terms that we can use to describe different elements and different groups in trees. On the left here, we have a really early tree. So this is one of the only figure, I think the only figure in Darwin's Origin of Species. And then we have a series of lineages from A to L here. And then going up, we have, I think it's every 100,000 generations or so. So we can see that certain lineages are successful, they proliferate, and they continue to the 14th generation or 14th 100,000th generation at the top, whereas some of them go extinct almost immediately. So this isn't really a phylogenetic tree, but it's I think one of the earliest points in the literature where people start using trees to define kind of biological or evolutionary kind of relationships. On the right, we have Ernst Haeckel's Pedigree of Man in the late 1800s. So obviously man is at the top, you know, followed by various different interestingly named groups of animals. So we have primitive mammals below man and the apes, and then we have things like jawless animals and reptiles and fish, for example. And then you have right at the bottom things like Monera. So Monera are what people used to call bacteria back in the day. So these are just all the kind of single cellular organisms right at the bottom here, and you know, this is quite kind of the arrogant way that man or Haeckel has done this, is we'll put man right at the top, which isn't really correct in an evolutionary sense, because all these animals are just as evolved as each other. You know, they're living today. So the way that we can infer trees, the way we can try and reconstruct evolutionary histories, we can use two different data sets. One of the ways is using morphology. So this is using fossils or anatomy. And practically speaking, for phylogenetics, how this works is that we code up hundreds of different characters based on those anatomical features, and then we can then determine, based on how many differences there are between those different character sets, we can try and determine how they're all related to one another. What I do, I use genetics, so I'm using things like DNA or amino acids. And the way this was done originally, we just kind of take a single gene and we'd look at the distances between different sequences of that gene found in different organisms. The way that the field has advanced in the last decade or couple of decades is that we now use a much greater proportion of the genome. So this is no longer phylogenetics, but phylogenomics. In order to do that, we're going to have to use sets of genes which are evolutionary orthologous to one another. What does that mean? I'll tell you in a second. But first, I'm going to show you some examples of what the data can look like. So on the left, we have an amino acid multiple sequence alignment. So every single site, every single column on the left represents a different homologous site. So different amino acids, we're going to see that, you can see that there's a lot of conservation in the middle, so that means that those GC amino acids there are probably doing something important, something to do with the protein structure. Some of the other amino acids are going to be different as you go all the way down. So that's going to be either genetic drift or perhaps those amino acids aren't as important to the protein structure. On the right, here's an example of kind of morphological way of looking at things. So this is the pentadactyl limb, which is kind of classic evolutionary biology point. Where we could use, for example, I don't know, the numbers of bones or the ratio or the angles of different bones, or the measurements of different bones, and we could code those into a morphological matrix. So I mentioned orthologous genes. So genes can be described, if genes share a common ancestor, they can be described as homologous, okay? And there are two broad groups of homologous genes. You have orthologous genes. These arise from speciation events. So the common ancestor of a group had one gene, its descendants had one gene. Those are quite useful, assuming that they have evolved in a kind of Darwinian fashion. Those genes can be used to infer species trees. But there are also genes called paralogous genes. So these have arisen through a duplication event. So at some point in an ancestral lineage, in an ancestral species, there was a gene duplication, which means every descendant of that ancestor had two copies of those genes. So if we then follow that through and infer a phylogenetic tree from those paralogous gene families, we get something like this thing on the right. We've got this alpha and beta paralog, where we have like a twinned kind of species tree event. Okay, so there are two copies in frogs, two copies in humans, two copies in mice. But if you were to infer a tree of them, each side is going to kind of mirror one another. It's going to represent these species-like relationships, but it's going to kind of double it. Now, the issue with paralogous genes, so this doesn't seem like such a bad problem to have. It just means you've kind of got two little species trees going on. Wow, that's loud. But what happens when there's gene loss after a gene duplication? So let's say we're going to lose, over time, various paralogous genes have been lost, and you end up with something like this. Okay, so on first glance, these look like orthologous genes. You know, we've got one copy in the human, one copy in the mouse, one copy in the frog. But if you infer a tree from that, you're going to get the mouse and the frog, who both retain their beta homolog, or paralog, are going to be more closely related to one another than to the human gene, which is obviously wrong. So we have to be really careful when doing our species tree reconstruction to make sure we're using genuinely orthologous gene families. So the way it works, the way we can use this multiple sequence alignment and kind of infer a phylogenetic tree is using a probabilistic model of molecular evolution. What does that mean? For example, if we take DNA models, some of the earliest ones would just say, okay, well, if you have a different nucleotide base, what's the probability? The probability is going to be equal that it goes to any other nucleotide base, okay? So A to T is the same as A to G, is the same as A to C and C to A, for example. As time's gone on, we've got more and more biologically realistic models. This also helps with the advance of computers as well, because some of these things used to take a very long time to run. And so the most complicated DNA model is the general time reversible model, where we have a different kind of probability on every single different combination of nucleotide to another nucleotide. And then for amino acids, we've got even more complicated models based on real kind of data. So that was a quick primer about phylogenetics. Now back to life, real life. So as I mentioned before, eukaryotes, we can divide life into kind of two functional groups, eukaryotes and prokaryotes, okay? But Carl Woese in the 90s suggested that actually based on some early phylogenetics, there are three domains of life, bacteria, the archaea, and the eukaryotes, as you can see in this kind of this famous tree of life figure in the top middle there. So that's what people thought. This is what the tree of life is for a very long time. But actually, modern phylogenomics has really changed our understanding of the tree of life. And we actually think there are two primary domains. There's archaea and there's bacteria. And then depending on which part of the eukaryote you're looking at, they are going to be more closely related to either the archaea or the bacteria. For example, the mitochondrial bits of the mitochondrial DNA is going to have a common ancestor with bacteria. Other parts of the eukaryote cell are going to have a common ancestor more closely related to archaea. So the actual tree probably looks something a bit more like this, where we have this kind of fusion later on with the archaea and the bacteria becoming the eukaryotes much later. But we're talking about LUCA, so we're looking at the common ancestor of all life. And it should be noted that there is a difference between LUCA and the origin of life. So life came into existence at some point, and then possibly a short or long time after that, we then get this common ancestor of all modern life, all cellular life. And this is what kind of a modern reconstruction of the tree of life looks like. So if we assume we're looking at the nuclear DNA of the eukaryotes, these are placed within the archaeal domain on the right here. On the left, we have the bacterial domain, and then the common ancestor of all these different things. So this is quite a nice figure that a PhD student put together, where he's put different pictures of all the different kinds of organisms. And we have, so humans, we're not even given our own group, we're called amorphia. So this includes fungi and includes amoebas. So this is right at the top there, you probably can't see very well. So yeah, really a very small part of the tree, and the eukaryotes are in green just below that. So how do we find out about LUCA? Well, you could take a kind of prebiotic chemistry approach. So some of you may have heard the Miller-Urey experiments. There are people like George Whiteflies and Nick Lane. So what these people will do is they will try and emulate early life conditions in a test tube, essentially. Sorry, not early Earth conditions in a test tube, and see whether or not we can get any biological molecules being created. So this figure here, which is quite cool, they tried to replicate early Earth conditions and see whether or not you can get biologically useful material from these kind of early Earth conditions, and they did, which is quite cool. But we take a slightly different approach. We're taking a bioinformatics approach where we're using phylogenetics to try and figure out what early life is like. So previous research into LUCA has come up with a whole host of different characteristics. Some people have said it was cellular, some people have said it was non-cellular, some people have argued about whether or not it even had a genome, the kind of environment that it lived in, what it was able to do metabolically speaking, where it was living, how simple it was. Is it a complex organism, or is it an incredibly simple organism? Is it even really alive? Is it, where did it live? Did it come from outer space? So there's been a whole host of research that we're really building on here with our work. Which is this, the nature of the last universal common ancestor and its impact on the early Earth system. So if anyone's really interested in this paper, it's all open access. It means anyone can click it, download it, and download all the data and have a play with it. So in order to find out about LUCA, can we use fossils at all? Yes, they are actually quite useful. So my background was actually originally in paleontology, and then I kind of switched over to the dark side of molecular phylogenetics. But fossils can tell us they are useful, even if we're not using them directly to inform on early life, because there are very few fossils that much older than, let's say, 3.4 billion years old, and those fossils that do exist don't really tell us much at all. Can't even really determine what organism they are. We can use fossils to calibrate our branch lengths. So I mentioned before branch lengths, they represent evolutionary distances. We can use geological information to kind of calibrate those evolutionary distances to real time. What does that mean? So we have our substitution model. We've got our gene sequences, we've got our tree, okay? So we have a molecular tree here. We then constrain using some fossils. We can calibrate various nodes on that tree, various bits of that tree. We can also use geological events to also constrain various things on this tree. And from that, we can then infer an order and an age for different characteristics of organisms. So in our paper, we wanted to date LUCA. Now, I mentioned before, in order to root the tree, you have to have an outgroup. The problem is there's not really an outgroup to life. So what we did was that we took gene families that previous scientists have suggested might have been in LUCA twice. So we think that LUCA probably had two copies. These are paralogous gene families present in LUCA. The reason this is thought to be the case is that if you take modern organisms, almost all modern organisms are going to have two copies of these genes, at least two copies of these genes. Actually, in the case of eukaryotes, they might have up to 6 copies of these genes, because if they have them in the mitochondria, they have them in the nucleus, and then they have them in the chloroplast, if they're a plant, that means they're going to have several different copies of these paralogous gene families. So what we did is that we used 5 paralogous gene families, rooted the tree between these 2 paralogous gene families, so we got this twinned species tree where we're going to have 2 descendant LUCA nodes, and then 2 twin species nodes to try and figure out how old LUCA was. And then we used a bunch of eukaryote fossils to try and calibrate that tree. So what do we get if we do that? Well, if we put it all together and we get this tree on the right where we've got eukaryotes represented in some cases 6 times, and we have these 2 LUCA nodes, and we have archaea and bacteria a couple of times, we infer an age of about 4.2 billion years old. So this really does kind of pushes back the origin of life to somewhere between 4.2 billion years and 4.5 billion years. So this is very soon after the Earth was formed. We think the Earth was formed 4.5 billion years ago. This is way before things like the end of the late Hadron bombardment, which some of you may have heard of. Way before things like photosynthesis. This really does push back the age of LUCA and the origin of life itself. So that's fun. That's a good result. But what was LUCA like? So we've got an age for LUCA, but how do we actually figure out, what was it living like? How was its metabolism? And for this, we need several things. We need a species tree. This hypothesis of evolutionary species relationships. But we also need a gene tree. We need gene family trees. And the reason for this is the kind of those orthologous gene families. They're generally not that informative about an organism's kind of ecology, its way of life, because the kind of gene families that are conserved across the whole tree of life, they're very much these fundamental biological characteristics, like all living things need them, for example. Like they're things like ribosomal proteins. So they're really not going to change much over the tree. Whereas the rest of the genome, these are going to be gene families that really represent what the organism is actually doing, what's the actual business of the organism. So, in order to do that, as I say, first we need a species tree. So this is a long way off from the ABCD tree that I showed you before. This is a tree of 700 prokaryotes. So this is 350 archaea and 350 bacteria. This is the maximum likelihood tree generated from 57 single copy orthologs. So these are 57 gene families that we think are present across the whole tree of life, or at least the prokaryotic tree. I haven't included eukaryotes for this part of the analysis because eukaryotes are complicated, as I explained. Don't worry too much about the methods there. That's just from the kind of, if there are any phylogenetics nerds in the audience, that's for them. Once we have our species tree, we fix that, okay? So we think this is how the species evolved. We're going to take that as truth going forward. It might not be true. It might not be correct, but we have to start somewhere. We then want to reconcile gene family trees. So we've got our species tree. Then we're going to infer from those 700 genomes, thousands and thousands and thousands of gene family trees, okay? And the reason for that is, as I say, we've got this species tree, this kind of scaffold that we think is how the species evolved, but we need to be able to figure out how the different gene families evolved. And because of things like gene duplication, which I mentioned before, or horizontal gene transfer, this is where different gene families can be transferred from one bacteria on one side of the tree to a completely different part of the tree, as well as gene losses. These kind of conflicts can traditionally make it quite hard to infer a species tree, but can be used here and reconciled with our species tree to try and give us an idea of when different gene families emerged. So we use this thing called amalgamated likelihood estimation, or ALE, and we can essentially use this to reconcile those different gene tree topologies with the species tree. And this, practically what this does is it gives us a number, it gives us a probability of each of those gene families being present all over the tree. So if we use that, then we can begin to figure out, okay, what was LUCA's genomic features like, So if we assume that the relationship between our gene clusters and modern organisms is the same going back the whole way through the tree, and we come up with a kind of predictive model based on our gene clusters related to the number of proteins and genes that an organism encodes, we estimate that LUCA had about 2,600 different proteins. And if we use the same kind of model to estimate genome size, we estimate a genome size of about 2.7 megabases. So what does that look like? What does that mean? Well, here are some examples of genome sizes of other organisms that live on Earth, or other bits of organisms. For example, the mitochondria, probably, ancestrally speaking, had a much bigger genome, but because its function is really quite limited today, it's just basically there to help make us ATP, the genome has been massively reduced. It's only 16.6 kilobases. Chloroplast, similarly, a bit younger than the mitochondria, has also undergone a bit of a loss in genome size. The smallest free living bacteria today has a genome of about 1.3 megabases. Your bog standard average E. coli genome size is around 5 megabases. So LUCA having a genome size of about 2.75 megabases, it's small, but it's not that small. This is considering how old, 4.2 billion years old. This is fairly big for a genome size that old. So I've lost some of the information here, but the largest bacterial genome today is around 14 megabases. As soon as we get to eukaryotes, genome sizes get way bigger. So the oak tree has a genome size of around 800 megabases. Humans have a genome size of 3 gigabases, and certain salamanders can have a genome size of up to 120 gigabases. I'm not entirely sure why that's the case, but it's certainly very interesting. Okay, so what do all our results mean? This. Okay, so we put everything together, we put all the gene families that we think are present in LUCA, we put them together, we run them through a database called the KEGG database, and we get a metabolic map for LUCA that looks like this. Okay, so the things in black, these are the gene families, the metabolic reactions, which we think are present in LUCA at a high probability, and high probability in this case means 75%. In grey behind is the reactions that we think are present at 50%. So that's kind of possible, but less probable than the highest confidence threshold. But what does that mean to a regular person? Because I have no idea what that means at the first glance. So this is our kind of reconstruction of LUCA. And reassuringly, our reconstruction, we do find things like ATP synthase. We find things like the ribosome. We find that it has, the nucleotide and amino acid synthesis enzymes. We have it. Interestingly, we find some evidence for things like sulfur reduction. We don't find evidence of nitrogen fixation. But what's really interesting is we find things like a hydrogenase, and perhaps really interestingly, we also find evidence for a CRISPR-Cas system. So some of you have probably heard of CRISPR-Cas9. So this is something which scientists today use to incorporate foreign genetic material into a different organism. So CRISPR-Cas9, for example, can be used in gene therapy. So if someone has a kind of genetic disorder, then we can use CRISPR-Cas9 to help treat that. But it's actually, ancestrally speaking, a bacterial antiviral system. So the way it works is that it basically allows the organism to take some of that genetic material from a virus and then protect itself against it. And we found support for 19 different CRISPR-Cas proteins in LUCA, which may suggest that already at 4.2 billion years ago, life is already fighting off viruses. It's already undergoing various pandemics. But where did LUCA live? There are various ways that you can tell where a microbe can live. You can look at certain marker genes, certain proxies, which you find across all organisms that live in a certain environment. One of the things we looked for was enzymes involved in ultraviolet damage repair. Okay, so an example of this is an enzyme like photolyase. We found a gene family called spore photoproduct lyase, which does a similar kind of thing. It was also annotated as photolyase in a different database as well, which is a bit worrying. And this is an enzyme that helps repair DNA after it's taken damage from UV radiation. We found this gene family with high probability, which could suggest that LUCA was actually living much higher up in the kind of water column. People have previously suggested LUCA was living in hydrothermal vents. This may suggest that LUCA was living very much near the surface and taking UV damage. However, when we looked at this gene family, we also found that it was present in things which don't produce spores. So spore photoproteolysis is involved in UV damage repair for bacterial spores, but we also found it present in things which don't live anywhere near the surface, things that are buried in the sediment. So this absolutely needs more work. It'd be really interesting to find out what this gene family is actually doing. We looked at another enzyme which is associated with hyperthermophilase. So there's a protein called reverse gyrase, which all organisms living at over 70 degrees, it has it. We found LUCA that had this with a high probability, but again, it's not a clear picture because reverse gyrase has a very complicated evolutionary history. It arose from a kind of gene fusion event, which is very hard to kind of disentangle using our kind of methodology. So we took another approach to trying to infer the optimal growth temperature of LUCA, and this was, equally ambivalent. We can't say one way or the other. So we put all this together. We put our understanding of LUCA together, and we get an idea of LUCA, which has to rely on abiotic chemistry. We don't think LUCA had the capability of doing nitrogen fixation, so that means it's going to have to rely on atmospheric chemistry, things like lightning, for example, to help produce biologically available nitrogen. Or it wasn't living alone, and it was already embedded in an early kind of ecosystem where other organisms are going to help produce some of the things that LUCA needs to live. Or if I think it is possible that our inference of LUCA could have lived completely autotrophically, and if that was the case, then certainly it would have helped kind of pioneer one of the earliest ecosystems. The metabolites LUCA was producing would have certainly been used in any other organisms living around the same time, which obviously there's no evidence for today because they would have gone extinct and there's no fossils for them. So any kind of organisms living alongside LUCA would be very hard to actually find out if they were living there or not, but it seems unlikely that LUCA was totally alone at that point. So the take-home message really, here we got the AI reconstruction of what LUCA looks like, which is completely mad. But yeah, previous inferences of LUCA, they vary, but we take this probabilistic approach and we can assign a kind of certainty based on our reconstruction. We estimate LUCA's age to be really quite old, 4.2 billion years. And then we find its genome is actually not, it's not this simple few hundred proteins. This is a complex organism that's evolved relatively swiftly since the formation of the Earth. And it possibly even had an early immune system and embedded in an early ecosystem. So with that, I'd just like to thank you all for coming and thank all my collaborators. Obviously, I didn't do all this myself. This is an international collaboration with people all over the world working on this. But I'd like to say a big thanks to my supervisors, Tom, David, and Phil, and also a massive shout out to Sandra, who really helped develop some of the computational models for the dating part of the analysis. She really helped kind of integrate this novel cross-bracing approach for the dating side of things. I'd like to thank Melissa and her team at Science Graphic Design for the cover image, cover image that we commissioned, as well as my funders and the press for really kind of spreading this far and wide. Really did take off. So thank you.
I'd like to remind you that we're being joined at this event by an online audience as well. So we're going to take some questions now, but when I select you to ask your question, will you please wait until you've got the microphone so that the people who are listening to the talk online will hear your question before Ed answers it. So, who'd like to go first? Oh, lots of people. Right.
Thank you. Yeah, thanks very much, Ed. That was a really interesting talk. So I have two questions, actually. The first one is, so where do viruses fit in with everything you've spoken about? And the second one is, in a general sense, so what does the results of your study imply for the origins of life itself? Is it possible that does the existence of LUCA sort of imply that that life had a single origin, or that there was only one abiogenesis event, and the other lineages, well, there were no other lineages? Or is it possible that there were other lineages but they went extinct? Or is it possible that LUCA might, like the eukaryotes, are derived from multiple lineages in this sort of fusion event? That possibly LUCA had a similar origin, and maybe there are multiple abiogenesis events that feeds in. So yeah, that's not a very well-defined question.
No, thank you. Those are really good questions. One of those is a lot harder than the other one. So viruses, where do they come in? It's a really good question, and I'm not a virologist. But it's my understanding that viruses have actually evolved independently multiple times over the tree of life. So I think they've evolved at least 6 times. And the kind of viruses that attack humans and eukaryotes are very, very different to the kind of viruses or phages which attack bacteria. So I think our evidence that LUCA had a CRISPR-Cas system does suggest that viruses were already alongside this point in time. In terms of where they fit in the tree of life, it's a good question. My understanding is that they kind of they come about so almost inevitably once you've got a kind of self-replicating system. But again, I'm not an expert on viruses, so someone to ask would be Eugene Koonin, who's a lot of work on the origin of viruses. The second question is what does LUCA really tell us about the origin of life? Hopefully it tells us a bit. So one thing that it does do is kind of constrain this time frame. Depending on what you believe about the origin of life, it really constrains the time frame for which life could have originated. So this means that life must have originated relatively rapidly after the formation of the Earth. So we think Earth formed at 4.55 billion years ago. At that point, it wouldn't have been inhabitable. And actually, a few million years later, we've got the formation of the Moon. So the Moon arose as from a massive kind of Mars-sized object, collides into the Earth, and we think that this is a kind of planet sterilizing event. So even if life had originated before that point, I think most physicists would probably agree that it wouldn't have been able to survive in the kind of molten mess that was left at 4.5 billion years ago, or 4.45 billion years ago. Sorry, 4.45 billion years ago. So this means that the origin of life had to happen very swiftly, and it had to evolve some really kind of complex kit. Things like ATP synthase, things like a ribosome, the cell membrane, the cell wall. Hopefully, what this work does is it allows us to kind of have an endpoint. It shows us hopefully a bit maybe what like the end of that line between the origin of life and LUCA is. And hopefully it allows maybe other scientists, maybe people doing prebiotic chemistry, to try and work their way backwards beyond the limits of what my field, phylogenetics, can tell us, which is LUCA. So hopefully that answers some of your question.
Thank you. That's very early, 4.2 billion years. So you're implying that the first mitochondrion was present at that time because the eukaryotic cell had ATP synthase? So
we think the first eukaryotic cell, which probably had a mitochondria, we think that's probably around 2 billion years ago. Okay. So the common ancestor of all mitochondria was actually a derived alpha proteobacterium. So we don't think alpha proteobacterium could be dated to 4.2 billion years ago. We think LUCA had an ATP synthase. So yes, it does mean that the origin of ATP synthase is incredibly early, but it doesn't have any bearing on the origin of eukaryotes. That happened 2 billion years later.
Okay, but can I just ask the relationship between the current mitochondrion and the current chloroplast. Do we now understand what that relationship is,
Yeah,
and
so
how and when it happened?
yeah, so that's a really good question. So in terms of the mitochondria, we think this is an alpha proteobacterial ancestor, and all eukaryotes, ancestrally speaking, had a mitochondria. So this, so the eukaryote mitochondria is, shares a single common ancestor in alpha proteobacteria. The chloroplast found in plants or Archaeplastida, we think happened a bit later. Can't remember off the top of my head the age of Archaeplastida, but people like Phil and James Clark here in Bath have done a lot of work on the origin of plants. This came from a cyanobacteria. So cyanobacteria evolved oxygenic photosynthesis, and this became involved in a similar way to the mitochondria by the common ancestor of plants. Interestingly, with the chloroplast, there has actually been multiple kind of events where little plant-like things have also been engulfed by other organisms, and there's actually a series of different kind of chloroplasts which really mess up your phylogeny. Things like, I think, brown algae and various other types of algae, maybe golden algae, is that a thing? Various different algae have actually acquired a chloroplast in a manner of different ways, and so they actually have a much more complicated evolutionary history.
Okay, but basically the chloroplast didn't derive from the mitochondrion directly.
No, it's actually on the complete other side of the phylogenetic tree for bacteria. So yeah, it came from a cyanobacteria, which is a type of pterobacteria, whereas the mitochondria is an alpha proteobacteria, which is a type of gracilocute. So these are basically completely different. So you may know these as gram-negative or gram-positive. These are the kind of the ways that people describe these bacteria and the bacterial groups in the past. So yeah, chloroplast, completely opposite side of the tree.
Yes, so if LUCA and the age of LUCA implies that life emerged so rapidly after the formation of the Earth, and implicitly so easily, does that imply there ought to be complex ecosystems in other parts of the universe quite commonly?
It's hard to know. Unfortunately, we've only got this one data point, so I think I'd love to know the answer to that, but I really can't. I can't say. Perhaps we had the kind of perfect conditions on Earth that gave rise to life, or perhaps if we explore wider in the universe, we're going to find life all over the place. But that's an impossible question. I don't know the answer to that. I wish I did.
Thank you. You said there was a free living bacteria with a genome half the size of LUCA. So presumably it must have lost either duplicates or lost functions. How can you be so sure it's a descendant of LUCA and not a surviving ancestor?
Okay, so that's a good question. Yeah, there's multiple parts to that question. So LUCA. So genome size reduction is something that it can happen quite a lot. There are I think the Minimal Genome Project they've come up with the actual minimum genome size of about 500 proteins. I think this is a synthetic organism. Okay, that's incredible. That's way smaller than 1.3 megabases. So genome loss can happen for a variety of reasons. I mentioned that LUCA had 2,600 odd proteins, but we don't know necessarily what the function of all these proteins was. Things can become more efficient over time. So maybe you've got 10 proteins, ancestrally speaking, that are replaced by one protein today. So we could see this kind of reduction as evolution, or as natural selection makes life become more efficient. Because bacteria and archaea, they actually have their genome sizes tend to reduce over time. They're very good at getting rid of junk DNA. So, I mean, we shouldn't really use the term junk DNA anymore, but eukaryotes, for example, they have really big genomes. Most of it, we're not really sure what it's doing. Bacteria, anything that doesn't really directly have a function to its immediate survival, they lose it. One of the reasons they can afford to do that as well is because it's very easy for bacteria to share genetic information with other bacteria. This is how things like antibiotics resistance and super antibiotics resistance occurs, because bacteria living near other bacteria which are completely unrelated to it can share DNA. I think that probably answered the question. How do we know that everything? How do we know that these early small genome bacteria living today descended from LUCA and not something else? That's where phylogenetics comes in. So we can look at the kind of gene families that these modern bacteria have. We can infer phylogenetic trees using those single copy orthologs that I mentioned. We can infer a tree. We can look at it. And we can see, okay, this thing lives, this small free-living bacteria comes out here in the tree, common ancestor with these guys, common ancestor with these guys, a bit like the way that I showed you through the human evolutionary history at the beginning. But basically, yeah, phylogenetics answers that question.
I'm just going to interject here that we do have a number of people listening to this talk online, and I'm addressing you now. If you want to ask a question, please type it into the chat of the Zoom webcast, and I'll pick it up in just a moment, and you can ask a question that way. But for the moment, do we have more questions in the room? There's one.
This might perhaps be a silly question, so apologies. You mentioned the fossil record of these very, very ancient forms of life. Are there any fossil records of anything that isn't descended from LUCA? Or is LUCA just everything we found is descended from LUCA?
I mean, that's a good question. So the issue with ancient fossils, so even those things that we recognize, and I say, these are stromatolites, and I say they're bacterial, we don't know they're bacterial. We know that they're life. We think that we're convinced they're life, but it's when you just got essentially a few kind of lines in a rock, the diagenesis, the metamorphosis of the rock over those billions of years really kind of erodes much of the kind of biochemical signal. It's a hard question to answer. I mean, if we found something, if we found a fossil that looked like life, we still wouldn't actually be able to tell whether or not it was directly related to LUCA. And this is actually an issue that plagues the entire fossil record. We don't know exactly what an organism, we know that there is ancestral organisms, and we know that there are stem lineages, fossils, which will look a bit like the common ancestor. But these fossils aren't going to necessarily be a direct ancestor. It's just going to be something kind of living along at the same time, if that makes sense. Not really. I think, well, okay, no, we can be practical about this. I think the oldest diagnosable fossils where we can really say, oh, this is an ancient bacterium of this type, would be ancient ancient cyanobacteria, where we know that they're performing oxygenic photosynthesis. We have the isotope record, and we can see that these things clearly were ancient cyanobacteria. That happens way later. That's I think the earliest one is about 2.5 billion years. So before that, we know that they're life, but we don't know exactly how they relate to LUCA. They could be they could be something living alongside LUCA and not necessarily a direct descendant of LUCA. Absolutely. That's good.
Perhaps I could ask a question following up on that point. The geology tells us pretty strongly that the oxygen content of the Earth's atmosphere was very low until just over 2 billion years ago. We're talking about maybe 1% of the oxygen that's present in the atmosphere today. And yet, what you're telling us is that the phylogenetic approach that you're taking on genomes indicates that we have an organism here that's 4.2 billion years ago. That's 2 whole billion years before there's much oxygen, which is nevertheless, it looks like there's rather complex metabolism going on here. What do you have to say to that?
Yeah, it's a good point. I think there was a period in time where we thought photosynthesis was one of the earliest energy metabolisms that came into existence. But actually, we don't think that's the case at all. I think the current belief is that the 2 photosystems involved in photosynthesis, so involved in oxygenic photosynthetic bacteria, evolved in somewhere along the cyanobacterial lineage. The taxonomy gets confusing because if you spell cyanobacteria with 2 I's, then that is not necessarily the case. Anyway, we think that cyanobacteria evolved oxygenic photosynthesis, and then a couple of other kind of weird groups of organisms evolved non-oxygenic photosynthesis, I believe. But actually, before the Great Oxidation Event, 2.4 billion years ago, before oxygenic photosynthesis evolved, it means organisms are going to have to rely on a different molecule as the electron acceptor at the final point of the electron transport chain. So we predicted that LUCA had, we think it might have had elements of kind of sulfur reduction, or we also found that it had a hydrogenase. But the way that we think LUCA was producing its energy was a kind of early kind of acetogenesis metabolic pathway. So we think that it's kind of hijacking like an ancient carbon fixation pathway and using that to create energy in an anoxic environment.
More questions from in the room? We don't have questions online, but here's a question.
Hi. I know you're working backwards to LUCA, but are you able to make any predictions about anything that's missing in the fossil record, all right, that may in future come to light from looking at this tree?
It's a really good question. So that was, I think, part of the idea towards the funding for this project was whether or not we could, which elements of these ancient organisms can we falsify using geological evidence? And the problem is we infer an age of LUCA is so old. There's barely any fossils around. There's barely any rocks from 4.2 billion years ago. Because as I say, the geology has just churned over so much. All this rock has been subducted and lost over the time. The current geological record. Do we expect to find anything in the future? Hopefully we're going to find some things that may reinforce our results or show us where we were wrong. That's absolutely possible. Hopefully that's the case, and hopefully other scientists can build on our work and say, okay, well, these guys are clearly wrong for this, for X, Y, and Z reasons. Because I'm just simply a phylogeneticist. I don't necessarily understand all the biochemistry involved in these really quite complex metabolisms and machinery that we've estimated to be in LUCA. Hopefully we can possibly see some basically just some earlier fossils of life that really would help reinforce this. Generally speaking, paleontology, if you estimate a group as a certain age, over time you're eventually going to discover a fossil that helps push it back even further. We're discovering more and more fossils all the time. So hopefully that's the case with early life. Hopefully we'll find some older stuff that everyone agrees is clearly life, but I don't know. Yeah, hopefully.
Thank you. There were recently reports in deep ocean trenches of electrolytic processes producing nodules which could conveniently produce oxygen. And that could push back the oxidation forms, using oxidation to much earlier. extent than previously.
Yeah, that was a really, really interesting find. I know a lot of scientists, there was a lot of kind of controversy at the time as well when that paper came out. If that's the case, yeah, we could begin to use, if we could find the origin of that, then that may push back, this early oxygen record. It wouldn't. I don't think this would necessarily affect our reconstruction, because all we did, we kind of just naively said, okay, well, based on the modern genomes that we have, we work our way back because we think that these gene families are present. Having said that, we did find a protein, and this may have been an artifact, but we did find a protein involved in one of the photosystems in LUCA. We've kind of assumed that was an artifact, but perhaps more evidence is going to come to light that could suggest something otherwise. I know various colleagues of mine in Bristol are working on the origin of photosynthesis, and maybe they're going to have more to say about that going forward. But yeah, it's a really good point. Because oxygen will make a big deal to life. It's a massive thing, and it completely changes the kind of chemistry of the ocean. So yeah, it is a space to watch for certain.
Do you personally believe there's enough time for that level of complexity to have evolved?
That's an interesting question. All the work done on all the molecular clock analyses done. So okay. On Earth. So to put it into perspective, I did a molecular clock analysis on LUCA a couple of years ago for another paper, and we removed the formation of the moon as a maximum constraint. If you do that, and then you infer LUCA's age, and without doing this kind of paralog stuff, you get an age of LUCA about 12 billion years old, which is, I think, completely mad. That's before our solar system. So I don't believe that LUCA existed before that, necessarily. Do I think that that life could have evolved in such a short period? I mean, it's still 300 or so million years. That's a very long time. And evolution could have been happening a lot faster back then. It's possible that replication could have been a lot more mistake happy. It's not going to be the kind of refined, perfect replication system that we have today. So it's possible that it's just relatively a completely different scale in these first few 100 million years. Do I personally believe? I don't know. I'm a scientist. I don't believe. I don't know. I can simply present the facts as I understand them. I think it's certainly possible, yeah. But I think as we go forward, we're going to get a better and better understanding of why and how these things could have came into being. So if you ask me in a decade's time, what do I think? My view undoubtedly will have probably changed, because the science will have changed, and our image of LUCA will have been refined. So hopefully we get some answers to that question. But I don't know. I don't like to use the word like believe, for example.
I must say I'm really enjoying this discussion. I think that what you've just said is a conclusion that I've been coming to listening to what you've been saying this evening, which is that if we accept the maximum age constraint of moon formation, late Hadean bombardment, and so on, there's not very much time left in which what appears to be a very complex LUCA has to evolve in. And so one possible explanation of that, which you yourself just suggested, is that actually early evolution went very fast. But if that's the case, what implications does that have for your phylogenetic analysis if you can no longer rely on a relatively constant rate of genome evolution?
So luckily, basically, as kind of computers have become more advanced and our models of molecular evolution have become more realistic, we can account for that. So we can account for changes. We can account for changes in substitution rate at different parts of the tree. And as we go forward, our models are only going to become more and more biologically realistic. So in terms of dating, the way this is done is using a model called the relaxed clock, which allows rates to differ over the tree. But in terms of genetic distances, we also have models that account for this as well. And also, not only does it account for different rates in evolution at different lineages, also at different sites. So rates can be compositionally heterogeneous. So they can differ over the different structure of a protein. For example, there are going to be really important parts of a protein which are very rarely going to change, and then there are going to be other parts of the protein that are going to be churning over every kind of few thousand years or so. So we can account for that. And actually, I did do some research on some of these marker genes to see whether or not they appear to be evolving at a different rate on the branch separating archaea and bacteria. And it didn't seem to be the case that the kind of proteins that we're using to infer these early species trees, or these deep time species trees, were particularly biased in any way. So it didn't look like, at least the kind of genes that we're using don't appear to be biased. But it's one of the big questions is, how has the rate of evolution changed over time? For example, if you look at something like a mouse or a bacteria, they evolve a lot faster than we do, thanks to a whole host of different reasons.
Thank you. It's pertinent to this discussion, but what do you think the ambient temperature was in this early evolutionary phase? I'm thinking about how fast evolution can take place. You don't want to burn it, but you could work a lot faster than it's happening now, I suspect.
As in, like, the environmental temperature.
What's the body temperature? What was the temperature of reaction in centigrade?
Okay, so as part of my master's, I looked at this, and we got a temperature, I think what our models suggested was that it was 250 degrees, which is obviously insane. Like, clearly, like life, I don't think the molecules that make up life can possibly work at that temperature. But there are two kind of schools of thought on this. So one is, if you look at gene content and you infer certain marker genes like reverse gyrase, and you think that reverse gyrase was present in LUCA, that suggests that it is a hyperthermophile, most likely, working at over 70 degrees. However, so that's looking at a gene content kind of angle. The other thing you can look at is genome composition. You can look at the ratio of different amino acids encoded by the genome. And if you do that, I think some French scientists a decade ago did this, Mathieu Gruson, I think, and Bastien Bousseau. They tried to reconstruct LUCA's temperature based on an ancestral state reconstruction of the amino acids present in LUCA's proteome. And their model suggested it was actually much cooler. They called it a mesophile, so probably around 40-ish degrees, much cooler than suggested. Our work isn't definitive in regards to the temperature of LUCA, so I don't know. I personally, even though our model suggested that it had reverse gyrase, I personally don't believe that it actually had reverse gyrase. I think it's just an artifact of how reverse gyrase evolved, and it evolved in such a way that it would look like it was present in LUCA based on our model. So based on that, I suspect it was probably cooler than 70 degrees, but anywhere between I don't know, 20 and 50 degrees is on the cards, I think.
I think I would just like to ask maybe simplistic or naive question, but it seems to me when I listen in that you're facing constraints in terms of your backtracking to a point in time. The Earth was created or formed at 4.7 billion years ago. You've got a relatively sophisticated LUCA. The question is, how did it evolve so quickly? With the amount of time you've got left, and so I think my question is really, if the universe is generally reckoned to be about 14.7 billion years old, does this open the possibility that actually LUCA wasn't formed on Earth, it actually came from somewhere else?
There are certainly some people that think that's the case. So what's really interesting to me is how this research has been picked up by people in the astrobiology research community. And I watched a YouTube video actually the other day where they suggested that this research, which pushes back LUCA to 4.2 billion years ago, has kind of changed one particular YouTuber's mind on whether or not life could have originated somewhere else in the universe. I personally, I don't know if this research is necessarily convincing enough to convince me that was the case. But I guess it's a possibility. The issue with that is that what's more likely, that life evolved quite quickly in the first 300 million years or so, or that life originated on another planet and then came to, or another place in the universe and then came to Earth and then evolved slightly more slowly. I don't know. Again, it's almost. It's an impossible question for me to answer, really. So, but it is a good one, and it's something that I think a lot of people are thinking about.
Yeah, this may be a bit of a silly question, but we talk about life and the beginning of life. But what was it really that made the tiniest thing that started life alive? You know what I mean? I mean, if we have, basically, that's why you might think people believe in religions, because they say, well, it's God that has given life to,
So I think, it's a good question.
Sorry, I was going to say we can find out, for example, what a tiny bacteria is made of. We could try to recreate a bacteria in the laboratory, put in all the elements, and yet, even if we put all those elements in, we cannot create a live bacteria. So what is it? Do you think that really life started as a fluke?
It's a good question. But it's a bit like asking, what happened before the Big Bang? So in my field, the earliest point we can really get to is something that's already alive. This LUCA, this common ancestor of extant cellular life. If we going beyond that is going to involve someone cleverer than me. It's going to involve prebiotic chemists, people like George Whitesides, people like Nick Lane, and hopefully they can kind of use the results that we came up with and try and push back slightly. I can think of other ways that you could possibly go beyond LUCA. You could look at those kind of those pre-LUCA paralogs that I mentioned. You could also look at perhaps the evolution of different. If you imagine proteins to be a bit like Lego buildings, for example, those Lego buildings are going to rely on certain bricks, and possibly we can kind of tease out, with the help of things like AlphaFold, the evolution of protein structure, perhaps we can tease out a kind of a simple chronology of when different proteins evolved, did they evolve in a certain order before LUCA? But that's beyond me. That's not my field. But yeah, understanding what came before LUCA is fascinating. But yeah, hopefully we can help answer that question. But I can't answer that question.
Well, really, nobody knows.
Yeah.
So one thing that's sort of concerned me in the light of some of the more recent questions is if the outputs of this model is being so driven by a few constraints or having to be fudged to work around what people would probably regard as common sense constraints, does that suggest there's something fundamentally wrong with the underlying model?
No, I don't think so. I mean, I wouldn't say fudged. I mean, we're talking about the origin of the Earth. You know, so we've I think sensibly put a maximum constraint on the age of LUCA to be the formation of the moon, because my understanding is the way that the Earth was formed, or the moon was formed, a massive Mars-sized object crashes into the Earth. This essentially, I think the current theory forms this thing called a synestia, where this Earth becomes this kind of dissolved donut shape, and then the moon condenses from kind of the outside of that. This explains the kind of geological makeup of the moon, and the rest of it becomes the Earth. If life already was on Earth, if life had came to Earth in some way, shape, or form, or evolved on Earth before that, it undoubtedly would have been sterilized by that event. So we're relying on life happening to land on Earth between in those kind of early few hundred million years. I don't see it. It's undoubtedly going to be a kind of naive approach, and there are things we're absolutely going to work on going forward. But that's just how science is, really. Like we're just trying to do the best with what we can. So yeah, I wouldn't necessarily use the word fudge, but this model is undoubtedly going to be wrong in some ways that we're yet to discover. But hopefully, as we go forward, we can find out what they are and improve going forward.
Actually, although we have another volunteer for a question there, I'm actually going to draw the discussion to a halt now, because I think that we could be here a very long time talking about this and probably going round in circles. What I would say in my concluding remarks is that Ed has very successfully got over the idea that this is a really crucial area that we do not really understand about the early evolution of life, and that he has described to us a way of looking at this, a rather new way of looking at it, which tells us that there are really important things that we need to find out. And I very much hope for Ed that this enables him to spend the rest of his career finding out, and we'll ask him to come back here again in a few years and tell us how he's got on. I think this is an absolutely fantastic evening tonight. This is one of the best BRLSI evenings I remember for many years, and so please let's all thank Ed for a really terrific talk.
Thank you very much.